I don't want to draw out the whole thing. I mean, it would just take forever. So I'm doing it this way. But if I were to ask you to draw DNA for me, and again, the chances are really high, this is what I want. It's not that. It's this. So if you know the base sequence of one strand, you automatically know the base sequence of the other. Because the base pairing is predetermined, it's preset. So, thymine, adenine, guanine, cytosine. Again, adenine always to thymine, cytosine always to guanine, period. If you know the base sequence of one strand, you automatically know the base sequence of the other strand.
So the way that we would do on the test is we would draw that out, and then where it says B, you would
No, you
like
can just put
A.
B. B would be okay. You're welcome to put in the A if you want, and then put T on the other one. But as far as I'm concerned, B and B would be fine. Okay, that would be okay. Now, there's a reason why adenine to thymine, guanine to cytosine. You know, it's not by accident. Okay, I'm going to erase here. Everybody okay here? Okay. I just kind of remembered, I gave you counter parallel for the way the strands go up and down in opposite directions. You also will sometimes see anti-parallel. Both terms are used. They mean the same thing. Two-lane street going in opposite directions. So anti-parallel, counter-parallel.
Same.
They're both used, yeah. I thought I'd better mention that. I can't remember which one the book uses. Okay, so what I want to do with this is to tell you that adenine you don't need to remember this. This is off the record. Guanine have a double ring structure. Cytosine and thymine have a single ring structure. So that's kind of off the record. So what I want to show you is, let's pretend that there are no rules. We're back on the record again. So let's pretend there are no rules, that anything can bind to anything. So I'm just going to put circles in instead of drawing out the rings. Again, just to make it quicker. So let's take adenine, so double rings, take adenine, and let's let her bind to adenine. Remember, we're saying no rules. So anything can bind to anything. Now we'll take adenine, let it bind to thymine. Thymine just has a single ring. Now let's take cytosine, single ring. Let it bind to thymine. Now we'll take guanine. Now let it bind to adenine. So if we take the other strand, you don't need to be a DNA expert to realize that doesn't look very good. It's not stable. It just looks bad. So now let's actually take the actual rules. So adenine to thymine, guanine to cytosine. So adenine double ring, thymine single ring, guanine double ring, cytosine single ring, thymine single ring, adenine double ring. You get the point. By having these rules, the second strand is always equidistant from the first strand. Again, you don't need to be an expert to realize that just looks more stable than the other thing does. Remember, we're talking about heredity, talking about continuation of the species and all that. We don't want mistakes. And this looks better. So that's why. Adenine to thymine, guanine to cytosine. What I'm waiting for is for somebody to point out to me what I left out. I omitted something.
Why is it going to,
Pardon me?
if it's just about the, I guess, the size difference, or the double
Yeah.
versus single, then
Got it. So what she's asking is, so if adenine is going to thymine just to keep the 3 circles, why couldn't we do adenine to cytosine? That would keep the 3 circles, and it doesn't work. Well, the reason, that's the part I left out. The reason is this. It turns out that guanine will bind to cytosine with 3 bonds. It's not a triple bond, it's 3 separate single bonds. Adenine to thymine will bond to each other with 2 separate bonds. Again, not a double bond, 2 single bonds. That's why adenine cannot bind to cytosine, because cytosine wants 3 bonds. Adenine only has 2 to give. So between this and that, adenine always to thymine, guanine always to cytosine. And on that level of high excitement, probably a good time to quit. Any last questions on anything? Okay, again, if you do have any, if you like to know your score, email me tomorrow, or I'll see you on Thursday night. And of course, we 6 o'clock class. So last time we, I know it was after the midterm and we were cranking all that stuff, but we did cover a little bit of the genetics, the structure. We covered the structure of the nucleotide, the structure of DNA. We talked about the base pairing, that's the term, base pairing. The adenine to thymine, guanine to cytosine. Any questions of that from some later occasion? DNA, and well, what do we do with it? There's actually 3 different processes that occur. The first of them is simply called replication. Replication. Replication is making new DNA. Replication is making new DNA. Think it through. When does that occur? When does the cell make more DNA? New life forms? You're close. New cells. New cells. When the cell is dividing. So for us, of course, mitosis. And bacteria don't do mitosis, as you know. So that would be binary fission. But either way, when the cell is making new cells, then you need new DNA. That's when you do replication. Now, it would not be for making new life, because that's when we're reproducing and when we're doing heredity. Replication is not heredity. It's just making more DNA. So, I'm going to draw the DNA this way, just because it's easier. Not just putting in any old sequence. The exact sequence does not matter. Put in anything you want. All that counts is whatever sequence you have, make sure the other one matches. Again, the proper base pairing. So whatever your first sequence is, just make sure your second one is correct. So in this case, see the top strand base pairs with the bottom strand. So adenine to thymine, guanine to cytosine. Sorry.
Thank you.
That's not enough, I can get it over.
No.
Anyhow,
That's good.
now the enzyme that does this is called DNA polymerase. a polymerase. Poly means many. You've seen that before. Polypeptide, polysaccharide. The ACE, the ASE means enzyme. You've seen that before. So the only new part there is the MER, the MER. MER means subunit. MER means subunit. So a polymer would be a molecule with many subunits, and really many repeating subunits. Many repeating subunits. The polymerase is the enzyme that makes the polymer. Okay? So a polymer is a molecule with repeating subunits. And the polymerase is the enzyme that makes the polymer. Now actually, we've done polymers before. We just never used the word before. So think back and you think of any molecules that we've done before during the quarter that have repeating subunits. Anybody else? Protein, exactly. Proteins, because proteins have amino acids. And even though the amino acids are not necessarily identical, but they don't have to be. They're all amino acids. So a protein would be a polymer. Now along the same lines, for example, glycogen. Now glycogen, actually the subunits are the same. It's all glucose. So lots of repeating glucoses. That's a polymer. So like I say, we've done polymers before, we just never used the term before. In this case, the polymer is DNA, and the subunits are the nucleotides. So DNA is a molecule with many nucleotides. See, that's your polymer. And the polymerase is going to make the DNA. Now, the DNA polymerase is an unusual enzyme. It's unusual. It's a composite enzyme. It's a composite enzyme. What that means is it has several smaller subunits themselves are enzymes. A composite enzyme is made up of smaller enzymes. It's similar in concept to the quaternary protein that we did a long time ago. Quaternary protein is a protein that has subunits, and the subunits were proteins. And the big example, if you remember, was hemoglobin. So the quaternary protein is made up of smaller proteins. That's what this is. There are smaller enzymes, two of which I want to give to you. One is helicase. And the other is ligase. Ligase. Both of those are part of DNA polymerase.
Can you repeat that?
Yeah, ligase and helicase are both enzymes by themselves, but they are both part of the larger enzyme, DNA polymerase. So again, DNA polymerase itself is made up of other enzymes, and thus a composite enzyme. So what's going to happen here? And there actually are other enzymes. I just don't want to give you too many, so just these two. What's going to happen is that the helicase is going to unravel the helix. Now again, DNA, the double helix, it's going to unravel, it's going to unzip the two strands. they're the same two strands. Again, I don't care about your sequence, whatever sequence, but just make sure the base pairing is correct. But see, the two strands here have separated. That's what helicase does. It's still connected down here. It's going to come apart later, but for right now, one thing at a time. So the two strands come apart, and then another part of the polymerase is going to come and pick up new nucleotides. Now, where from? In the cell. You have heard me say on other occasions, the cell maintains storehouses and warehouses filled with raw materials. Maybe amino acids, maybe fats, maybe who knows what. It could be anything. So there are other nucleotides just simply being stored in the cell. Where do they come from? Why we eat. We eat, the food gets broken down, we get raw materials. Okay, so in this case, the other part of the polymerase will come pick up nucleotides and bring them back.
Is that the function of the ligase?
No. Just different part of the polymerase. And again, your base pairing. So adenine will bind to thymine, thymine to adenine, cytosine to guanine. Ligase comes later. So we have three different nucleotides. They're all bonded to the first nucleotides, but they're still separate nucleotides. This is where the ligase comes in. Ligase will sew together.
Is there an unique?
Well, if you want some more names,
Well, I wanna know what to be able to
just
call
call it
the
polymerase. Call it polymerase.
polymerase.
But I'm not going to be asking it of you. I'm not kidding. Call it polymerase.
Okay, so
I'm trying to avoid drowning people with names.
I'm just trying to.
So just polymerase. So polymerase, the rest of the polymerase will go and pick up nucleotides and will bring them in. So just saying polymerase will be fine for that.
Okay.
So again, the helicase is what separates the strands, again, and zips them, unravels, whatever. The other part of the polymerase will bring in new nucleotides. The ligase will sew them together. So now you have one strand, or at least a new strand.
Is this going on the far end of